Hi, everyone. Welcome to our cerebellar seminars. Today, as our guest, we have Jean-Jacques Orban de Xivry, JJ, as he's known to his friends. JJ received his PhD in computational neuroscience at the University Catholique de Louvain in Belgium, where he was mentored by Philippe Lefebvre, working on eye movements and applications to clinical populations. He went on to do a postdoc with us here at Johns Hopkins, where he used non-invasive brain stimulations, TMS and TDCS, to investigate the role of the motor cortex during adaptation. Subsequently, he was appointed professor at the Department of Movement Science at KU Leuven in Belgium, where he has had a very successful decade or more studying aging and neurological diseases and their effects on motor control and learning using a variety of approaches, including behavior, brain stimulation, EEG, and MRI. JJ, it's very good to have you.
Thank you, Reza. Thank you for having me. So today I'm going to talk to you about a project that is being run in the lab for the last 3 years now. So we are in the stage of writing all the papers. So this is all unpublished data that I'm going to show you today. Um, and if you look at the literature on aging, you can conclude that, where's my mouse? Okay. You can conclude that everything becomes worse with aging. I mean, basically, if you take a paper on aging, there are things that go backwards. And I'm actually not very happy with that thing because if you look around you, you could maybe find somebody like Joanna Kaz, who was the oldest gymnast in the world. On the video, she was 91 years old, and she could do gymnastic tricks that I've been unable to do all my life. So quite astonishing what she can do. Um, and I think this shows that I mean, it's not possible that, looking at the literature, that everything becomes worse, but then some people, older people, are
Sure.
still able to
I'm
do
gonna
things
hop
like
on
that.
to
And um, sorry, I heard something. Um, and that's also in the lab. We've also found similar things. So the first study on aging that we did was the looking at implicit motor adaptation. That was a study of Boon van der Voort, my first PhD student. And he studied implicit motor adaptation in a group of old and young participants. And you have here the implicit adaptation on the y-axis with respect to trial numbers. And this is how the adaptation looked like for the group of young people. And you could see more adaptation in the group of older adults than in the group of younger adults. And this and many other studies that existed at the time already really shows that implicit motor adaptation is not affected by aging. If anything, it becomes better. Now, in some studies, it's larger. In other studies, it's not different, but at least it's not deteriorated. And that was really the start of, for me, of a larger question about, okay, why is that? What is happening with um, this task that where older people are not worse than younger people, as you can find everywhere in the literature. And so we know that this implicit motor adaptation is a cerebellar-dependent process. And so you have here a very small part of the cerebellum with, for those of you who don't know, you have the Purkinje cells, which are the wider discs here. You have the molecular layer on both sides and the granular layer here in the beginning. And the question is really, okay, is cerebellar function resilient to cerebellar degeneration? And I'm gonna make this even more explicit. So let's suppose that this part of the cerebellum is actually involved in the control of arm movements. What would happen if you lose one Purkinje cell? Would that affect the control of the arm? Or maybe two Purkinje cells, or 10? And this is really relevant because we know that with aging, there is a 40% decrease in the number of Purkinje cells in the anterior lobe of the cerebellum, which is mostly responsible for the control of our movements. And so the question is whether this change in the structure, that is true, impact the function. And there are actually two hypotheses um, in the literature about this. There is what I will call the structure hypothesis, where it is stated that if there is a loss of Purkinje cells or a change in the structure of the cerebellum, it will directly affect the control of the arm movement. And that's, for instance, the Bernard and Seidler paper in 2014. And I saw that Jessica was in the audience. So I hope I'm doing her justice here. Or there is the reserve hypothesis that has been more recent, where people say, well, you know, there can be change in the structure of the cerebellum. It actually not directly it will not directly influence the function. And there are a number of papers in the recent years that are claiming that the reserve hypothesis might be a good explanation for the role of cerebellum in aging, but not only aging, also, for instance, for Parkinson's disease, that the cerebellum could serve as a reserve, so kind of as a backup for other function. Okay, so we wanted to dissociate between the structure and the reserve hypothesis. So two of my PhD students, Anda de Wit and Anouk Matthijs, did a very large experiment to try to see which of these two hypotheses was true. So they recruited a group of 50 young adults between 20 and 35 years old, a group of older adults between 55 and 70 years old, and a group that's 80 of them. So it was a very large group because we want to be able to do correlations as well. And then a group of old adults, so people above 80 years old, which is quite rare in the literature. And there were 30 of them. Another important aspect of this project is that we didn't focused on on a single task, because we know that the cerebellum has actually multiple functions. So we really wanted to investigate as many cerebellar functions as we could. And today I'm going to present you 7 different tasks that are all linked one way or the other to cerebellar function in many different domains. And so our idea is for each of these tasks to see whether aging impacts the performance on a very specific component of the task. Because in all tasks, and that's the only intersection between all these tasks is the fact that the cerebellum is implicated in it, and that we can really measure the contribution of the cerebellum via a specific outcome. For each of the tasks, we have on one hand a general sensory motor measure, which is basically the overall performance of the participants on the task that reflects the working of the cerebellum, but not only. And then we have cerebellum-specific measure, where we know these parameters really reflect the contribution of the cerebellum to the task. And how do we know that? Well, either we know that this specific parameter is affected by cerebellar degenerations in patients with early cerebellar degeneration, so not due to age, or there is a theoretical explanation that there is a need for an internal model to solve that task. And if we take for granted that the cerebellum is a place for the internal model, then that would be why the cerebellum should be implicated. So let's turn to our first task now. And the first task is, of course, a motor adaptation task. So we use the task Clamp feedback task that was developed by Rich, Ivory, and Ryan Moorhead. So on the baseline, it's a typical reaching task. So you don't see the hand, you only see the red cursor, and you have to bring your hand from the starting position to the target. But then after some time, we introduce a perturbation. So now the cursor will be deviated away from the hand's direction. And you don't see the hand, you only see the cursor. What's specific about the task is that it's not a typical visual motor rotation. The cursor will always go in the same direction. So here, 30 degree or 45 degree to the left of the target. And so after some time, the hand will shift in the direction opposite to the rotation of the cursor, but the cursor will still go towards in the same direction so 45 degrees to the left of the target. And we tell the participant that we introduce this perturbation. We tell them to just neglect it. Just try to bring your invisible hands to the targets. That's the instruction. And we make sure that the participants understand that. Ryan, in his paper, has shown that cerebellar patients show less adaptation than healthy controls in this specific task. So we know that this adaptation is linked to cerebellar function. So what do we have in our data? We have the hand adaptation on the y-axis with the number of trials. And here we have the amount of adaptation is a cerebellar-specific measure. And basically what you can see here is the average for each of the three groups, the young in blue, the older group in orange, and the old group in purple. And basically, as you can see, there's really no difference in adaptation between the two groups. There are small differences, but that falls within the variability. So we can do the statistics. Here we don't see larger adaptation. In older participants, it's slightly higher in older adults, but not significantly so. And so we see very little difference for this in this experiment. So basically for this first task, which was implicit adaptation, this seems to favor the reserve hypothesis because we see no difference between young and older participants. The second task is also upper limb task. It's a sensory attenuation task, a force matching task. This is a task that was developed by Daniel Wolpert that you've probably heard of. I'm gonna explain quickly how it works. So first, there is a force perception phase in the task. So there is a small motor with a lever, and this will apply a force on your finger. And the idea is that you experience this force and you have to reproduce it. And there are two ways to reproduce it. In the first way, that's what they call the slider condition. You use a small button to control the motor such that the motor will activate the lever to press on your finger. That's an indirect condition, a slider condition. And when you do that for different level of force, this is the presented force versus the force that is reproduced by the participant. The dashed line here represents the ideal performance. And you can see this is for a group of young participants that the performance is quite good. Now, there is another condition which is called the direct condition. In this case, instead of using a button, the participant will directly press on the other finger on the lever to exert a force on the other finger. And so this is supposed to cause sensory attenuation because you are pressing on your own finger. Your brain will anticipate how much force is being produced, and you will be less sensitive to the force that you produce yourself. And this is exactly what we do because people are less sensitive to the force that they produce themselves. They will actually produce more force too much. And so the difference between the slider condition and this direct condition is the overcompensation. That's a sign of sensory attenuation. And so we repeated this experiment with a very similar device that had been done by Noam Wolpe also. So we wanted to reproduce this. And so first, the performance in the slider condition is a general sensorimotor measure for us. It represents how good people are in general at reproducing a force that they have experienced. And this is our data. So you have the target force here. We only use 4 levels of force and the match force. The dotted line here is the ideal force compensation. You can see that the younger participants are quite close to what they should do, but that there is a small underestimation of the force by the group of older old participants. And we can do the statistics, and the group of younger, the error or the slope or good people can scale the force with the target force is represented by the slope of this line. and the slope is slightly higher for the young participants than for the older or older old participants. And so there is, for the general sensory motor measure, we have a small effect of age, but that's not a cerebellar-specific measure. So the cerebellar-specific measure is what we measure in the direct condition, because only if you can predict the sensory consequences of your movement, you will be able to attenuate the feeling and to show overcompensation. And this is the cerebellar-specific measure, and this is the results that we have. So the dashed line here, those are the lines from here. And then you have the data from the group of young in blue. There seems that they are very similar to what you see here. And then you see a higher force for the older participant and even a higher force for the older old participant. So basically, the older you get, the more force you produce in this direct condition. To compute the overcompensation, we do the difference between what the force that they produce in the direct condition with the force that they produce in the control condition. And this is what we observed here. We actually saw no overcompensation in the group of young participants. And we have an explanation for that. It's just the way we did it doesn't lead to a lot of overcompensation in that group. And then for the older participants, we saw more overcompensation for the older and older old group. So this again suggests that these older participants, they could really predict the sensory consequences of their movement, and they could attenuate the feeling. And so this means that the cerebellar function, again, is intact in this group, which is consistent with the reserve hypothesis. A third task that we looked at was the interjoint coordination task. So this is a very simple task where people are in this posture and they have to make a very simple flexion movement. When they do that, due to the laws of physics, this will create an interaction torque on the upper arm. So even if you do a pure rotation of the lower arm around the elbow, that will create a motion for the upper arm. That's the laws of dynamics. And so that means that if the brain wants to stabilize the upper arm, it needs to activate the shoulder muscle to counteract this interaction torque. And we know that young participants, they can do that. That has been reported by many people, including Paul Gribble. And we also know that this interjoint coordination, compensation for interaction torque is a cerebellar-dependent process because it is mostly absent in patients with cerebellar degeneration. So could we find this activation of the shoulder muscle to counteract the interaction torque? Yes, we did. Oh, sorry, I have another. I'm going to show that and I'll go back. Yes, we did. So here in blue, we have the activation of the shoulder muscle. That's the pectoralis for flexion. And we have activation of the arm muscle in gray. And we can see that the activation of the pectoralis occurs earlier than the activation of the biceps. And this is for the group of younger participants, but we observe exactly the same for a group of older participants. So we do have this anticipation, this compensation for the interaction torque that works as well for the older participant as for the younger participant. And we can do the statistics on that, and we see no significant difference between the two. So here the color bar is always the timing of the activation of the shoulder muscle, and the gray bar, the timing of the activation of the biceps. And we've done that for the extension as well, and it gives exactly the same results. But this very precise activation of the shoulder muscle is not sufficient to actually maintain the general performance. If we look at the shoulder angle, so the shoulder angle, the shoulder is supposed to stay at 60 degrees, but we see that in all those groups, there's a small deviation of a few degrees, and this deviation is larger for the group of older old than for the group of young. So even though the anticipatory mechanism to compensate for the interaction torque seems to be present, they're not sufficient to maintain the general performance. So we see that the cerebellum works, but there is something else. Early on, we actually see when the interaction torque is compensated at the beginning, we actually don't see any difference between the three groups, but later on in the movement in a later phase, that is probably more based on the sensory feedback there's a difference that starts to emerge. Okay, so that was our third task, which was the interjoint coordination task. And as you can see, so far, I've only talked about tasks from the upper limb, but we wanted to be very general. So we also tested other type of tasks that are not only based on the upper limbs. And so the next task is an eye movement task, which is actually the task that I used during my PhD thesis. But in my PhD thesis, I only tested young participants, and here we tested also older participants. So how does the task work? It's a task where there's a lot of anticipation and prediction involved because you have to track with your eyes an invisible target. And how does it work? It works like this. There is a moving point that disappears and then reappears. Here I show you many trials, but it's actually one trial at a time. So the point moves, disappears, and then reappears. And you don't see the occluder. The occluder is invisible. Here I just illustrate the task. So what is the typical performance in such a task? So here you have the position of the eye versus time and the velocity of the eye versus time. In black, you see the position of the target and the velocity of the target. On the position trace, the thick traces are the saccades, and the thin traces are the smooth pursuit. And it's the other way around on the velocity trace. The thick trace is a smooth pursuit eye movement, and these really huge peaks in eye velocity are the saccades. So to perform that task, we use a combination of saccades and smooth pursuit. And what's interesting is that if you look at several trials from the same participants, you see that there is some variability in the contribution of smooth pursuit eye movement and saccadic eye movement. So here is another trial where you can see that the saccades here are larger, and then the smooth eye velocity is lower. Here, another trial from the same participants where the saccades are smaller and the smooth eye velocity is higher. And here, even a more extreme case where there is only one saccade and the smooth eye velocity remains high. So, and that's all trials from the same session, from the same participant. So even from trial to trial, there is quite some variability. And the question is, okay, how is this organized? Is that random? What the size of the saccades and of the smooth eye velocity are? And the answer is no. So how do we quantify this? For each trial, we can compute the contribution of the saccadic eye movements and the contribution of the smooth pursuit to the eye displacement during the phase when the target is invisible. So this gray phase here when there is nothing on the screen. And so we can plot one versus the other. So we have a graph here. This is the saccadic displacement. So this is how much the eyes move with, thanks to saccades when the target is invisible. And this is how much the eyes move during the occlusion thanks to smooth pursuit. And if we take the 4 trials that I showed you before, they fall almost on the line of perfect compensation. So 23 degree, that was the target displacement during the occlusion, so when the target was not visible, and so this should be the perfect compensation. These are for 4 trials, and this is for all the trials of that participant. And so we can basically look at this relationship, and we can either compute the correlation or the slope of the regression line here. With if the slope is -1, it means a perfect compensation between saccades and smooth pursuit. If the slope is different, it means that the compensation is not perfect. Why do we think that this is linked to cerebellar function? Well, we think it's linked to internal model function because there's actually nothing on the screen when this coordination of the saccades and the smooth pursuit happens. So it might be that the pursuit system is controlling the smooth eye velocity of the eye, and then a reference copy is sent to the internal model in order to predict what the smooth eye displacement will be. And this is also sent to both the pursuit system and the saccadic system, because there is no other way to do, because there is absolutely nothing on the screen during that second. So the coordination between the two cannot be due to visual feedback, because there is absolutely nothing on the screen. The coordination happens in the absence of any visual feedback. So the correlation should be due to the fact that some information is shared, non-visual information is shared between these two systems. So this was this is one of our cerebellar specific measure for this task is the slope of the regression line. And we expect that a slope of -1 means perfect compensation. But there is another outcome that could also be cerebellar dependent. And this is what we can observe before the onset, before the target starts to move. So here you have the target starts to move at this point, and you can see that the eye velocity is not 0 before the target starts to move. And this is called anticipatory eye velocity. Because we repeat the same target motion over and over, participants learn the timing of the task, and they are able to start the motion of the eye before the target motion starts. And we know that this anticipatory eye velocity are also absent in patients with cerebellar degeneration. So that can be also a cerebellar specific measure. And then we have a global measure, a general sensory motor measure, which is the smooth pursuit gain, so the eye velocity at the end of the visual tracking when the target is visible. So we're gonna look at how much these 3 measures differ with aging. So first, the slope. And here we can see that the slope for the 2 age group, we didn't test the old participant here because doing eye tracking in people above 80 plus is very, very challenging because they don't open the eyelid properly. And so the tracking is very bad. So we didn't do it. We only have a group of young and older participants here. But here we did find an effect of age on the slope. So you can see that on average for the young participant, the slope is -1, which means on average there is perfect compensation between smooth pursuit and saccades. Of course, there is quite some variability across the participant. But for the older participants, this difference, the slope on average was mean 0.7, which was significantly lower or closer to 0 than for the young participant. If we look at anticipatory eye velocity, so the eye velocity before the target starts to move, here we saw absolutely no difference between the 2 age groups, and that's also a cerebellum-specific measure. And if we look at then on the general sensory motor measure, which is the smooth pursuit eye velocity, the steady state smooth pursuit eye velocity, or the eye velocity gain, in this case, we saw again a difference between young and older participants, which is well known. The gain in smooth pursuit is lower for older people than for younger people. Okay, so again here, how we can conclude is that, well, this is mixed findings for this task. One of the parameters supports the reserve hypothesis, and the other one might be consistent with the structure hypothesis because there was a difference in slope. After the eye, we move yet to another thing, and that's this time we looked at speech. Because because we know that we can do adaptation of the arm and we can also do speech adaptation. So we, with the help of Ben Parrell, we set up a speech adaptation task. And the idea is quite simple. We ask participants to repeat the word bed, and we modify the auditory feedback such that they hear beat. And that will make the participants adapt what they are actually saying. And we can track the frequency of this vowel here in order to look at how much they shift what they say with respect to their auditory feedback. It's quite a boring task because you have to repeat bed many times. But that's what we did. And these are our results. Those are the unnormalized F1 curve. And just to show, I want to show you this because you can look at, even at baseline, the frequency of the A vowel was quite different between older participants, older participants, and young participants. So here, that's a global or general sensorimeter measure. Just the baseline frequency is already different. But we can normalize that, but at least it shows that, again, the general sensorimeter measure, there is an effect of age on this frequency. Now, if we try to quantify the adaptation itself, we can normalize everything with respect to the baseline and then look at how much the frequency shifted with the perturbation. So the perturbation starts here. This is the frequency of the first 4 months. And we measure adaptation here at the end. And what you can see is that for the group of young and old participants, on average, they're very similar. If anything, the older participants are slightly better than the younger participants, and the older old group seems to be lower. Now, the thing with this task is that it's actually very variable. So if you look at the amount of adaptation and how much variability there is between the different participants, there is huge variability, which means that basically nothing is significant. So we do not have evidence that there is an effect of age on this task, even though there seems, if we only consider the mean, there seems to be an effect. At least if we compare the young group and the older group, we really don't see an effect. If anything, the older group are larger. What the actual conclusion is for the older group, it's very difficult to say. It seems to be lower, but there are also those 2 points that are much lower. So yeah, it's hard to say exactly what it is, but there is no clear effect of age on that measure.
Sorry, just quick question.
Yeah.
What do the error bars mean? They SEM 95% standard deviation.
They are confidence interval. No, they
95% intervals.
Yeah,
okay.
yeah, that's what I would say.
Thank you.
Not sure 100%. I would need to look. I can check. Okay, so that was the speech adaptation. So even though I would agree that for the old group, there is some uncertainty, that doesn't seem to be, it seems to be consistent with the reserve hypothesis more than with the structure hypothesis. The next task is a mental rotation task, which is a task that was tested by Sam McDougall and Rich in patients with cerebellar degeneration. So Sam sent us the code to run the task, so that's what we did. So the task is quite simple. You see a letter on the screen, and you have to answer whether the letter is in the correct position or whether it is flipped. So here, that's the correct letter, but there's also some cases where the letter can be flipped. And we look at how long it takes for the participants to answer this simple question. And Rich and Sam found that, and they did not find it, it was known that the amount of rotation, the reaction time correlates with the amount of rotation. So the larger the letter is rotated, the more time it takes to answer. And that's a linear relationship that's been found already a long time ago. Sam and Rich reproduce it in a group of control. And what was also very interesting in the paper is that if you look at patients with cerebellar degeneration, you can see that the slope is larger. So that's why they quantify here. This is the slope. So how many milliseconds of reaction time do you have per degree of rotation? And this slope was larger for patients with cerebellar degeneration than for controls. And so they had more difficulties to perform the mental rotation. So we repeated this task in our participants, and this is our data. So here you have the reaction time, and you have the rotation. We didn't test as many rotations as Sam and Rich did, because we had so many tasks that we also needed to make some choice. So this is the young. In the graph, the dashed trace is the data from the young participants that we just offset it to look at to illustrate the difference in slope. So there is a very small difference in slope here and a larger one here. If you do the statistics, there is actually no difference between the first two groups. So the people between 55 and 70 years old had a very similar slope as the people between 18 and 30 years old. The slope is indeed larger with respect to the young participants for the group of older old participants.
Why
And
is the slope? Why is the slope a more specific measure than the offset? Why do you
So
think?
the slope is a more specific, because that's what they showed here in the paper, is the idea that you have to do this mental rotation, that it's the mental rotation that is a cerebellar-dependent process, which is what they show. There's also a shift in the reaction time, of course, but in the paper, they argue that it's this continuous update of the mental representation that is cerebellar-dependent.
Right, but the data in the middle panel make it pretty clear that the effect size for the offset is greater than that for the slope. Looks like.
Yeah, there is definitely an
that would
but
suggest that
they have other experiments where the difference is not as big here. Yeah.
Right, so that would suggest that the offset would be a more powerful, statistically more powerful measure.
Yes, but the offset is probably affected by many other things than just this continuous update of mental representation. And
Okay,
there's another
thanks.
question in the chat about the R. Yeah, so of course, if you have a symmetric letter, then it doesn't work because you can't do the task. You don't know whether it's flipped or not. And we use here it's with a R, but we use different letters. So Rich can correct me at the end if I misinterpreted his study. All right. So here for the mental rotation, we also have mixed results. We don't see an age effect with respect between the young and older group, but only with the oldest participants. And the last task is also not really a motor task. I mean, it's a motor task in the sense, but it's a timing task, especially. There's lots of experiments and papers on the fact that the cerebellum is involved in rhythm or in motor timing. And so we repeated a typical study here where participants hear a beep. Here it was every 600 milliseconds, and they have to tap in synchrony with the rhythm. And then the beep stopped, but they have to continue for 31 trials to tap at the same rhythm. That's typical rhythm task that was used, and we know that patients with cerebellar degeneration are impaired in some specific components of this task that we can measure. So if we look at the tap interval, there seems to be some difference in the mean here. If you look at all those trials, and there are indeed, but it's much smaller than the variability between the different participants. So that's what would be our general sensorimotor measure. But if you look at the variance, so it's possible to fit a model where you have a motor variance and a clock variance, and the clock variance is increased with cerebellar degeneration, which we didn't find any change with age of the clock-related variance. So there, it's a bit complicated to explain how you obtain that, but there are many papers on these clock-related variants. So it's possible to estimate it with a model, and this is the outcome of that model. So there's the standard deviation of that is due to the time processing. So the model really makes a difference between the variance in the motor component and the variance really in the clock component. And so this is the cerebellar-specific measure, which again is not different between young and older participants. So within all the tasks, as you can see, there's quite an array of results. Most of the tasks support the reserve hypothesis. There are either one outcome for the anticipatory eye tracking or for the mental rotation, the group of 80 plus. That is where we see some differences, but most of the tasks actually are consistent with the reserve hypothesis. But now maybe we know that when we recruit all the people in the lab, we know we have a biased sample because we get the best older people that are very healthy. I'm on sport campus, so people that like doing sport as well. So maybe they do not represent the whole population. So we're wondering, okay, do these people have cerebral degeneration? So we also did a structural MRI with them, and we measured the volume of the cerebellum. And we did see that there was a difference in cerebellar gray matter volume normalized by the total intracranial volume with age. So definitely there is a loss of cerebellar gray matter with age between our groups. So we indeed have a change in the structure, even though we don't see a direct impact on the function. What was interesting is that when we look at these results slightly further, and instead of normalizing the cerebellar gray matter by the total intracranial volume, as is mostly done, but if we normalize with respect to the total brain gray matter, there we see an increase with age. And what this means is not that there's more gray matter in older people, but that actually the decrease in gray matter in the cerebellum in our sample seems to be lower than the decrease of gray matter in other brain areas. So relatively, there is more cerebellar gray matter in older people than in younger people, because potentially the loss of cerebellar gray matter is smaller than the loss of gray matter from other regions. And we look in the literature, and this seems to be a pattern that has been found by other people, but because it's the cerebellum, many people don't really pay attention to this. So this seems to be a finding. It's not only specific to our sample. To conclude, I think I showed you that even though we had a change, an age-related change in the structure of the cerebellum, we did not really find an age-related change in the function of the cerebellum. And of course, we can argue for every of the task whether this is really cerebellar function that we measure. I think that's based on the literature. That's how we selected the task. We can discuss for all of them. I think if for all of them we come to the conclusion that they have nothing to do with the cerebellum, then maybe we need to revise our theories of motor control. So basically that means that cerebellar functions are largely resilient to age-related structural degeneration. And this idea of cerebellar reserve is this possible, but also we see that this is not sufficient to maintain global motor performance. Even though we saw that the cerebellar-specific measure did not change with age, most of the global performance measure did change with aging. So it's not the solution to all the problems, but at least it's interesting for me to understand what does deteriorate and what does not deteriorate with age. And this is really the work of Anda and Anouk, who are both in the last year of their PhD. And so the papers, they are working on all the papers linked to this study. Thank you for your attention.
Well, JJ, that was the tour de force. Thank you so much. Please, if you have any questions for JJ, just go ahead and raise your hand. I will call on you. JJ, I wonder if you can go back to the rhythm production slide. And I was a little confused about what was being plotted. So you were plotting intervals on the y-axis, and of course a smaller interval means that they're going faster.
Yes.
Why did they go? What's going on there? Why do they go faster when you remove the reference?
I don't know. I think it's the tendency. People anticipate that a little bit too much. I mean, this is a task that Rich has studied extensively, probably knows better than me why it is so. But we see that they are going slightly faster in all the groups. What is the reasons that I don't know.
All right, Shlomi, please go ahead.
Thank you, AJ. Beautiful work. I love it. My first question is, you started with saying that you have such a big group so that you can look at correlations. And I'm wondering, where are those? So specifically with the MRI analysis, if you can show us any correlation within the older groups between their MRI measures and their performance, that would be the full proof that you will nail it.
Yeah, so we haven't done them yet because we still looking. That's not gonna be for the first paper because that's gonna be too much. We haven't done them yet, so I can't answer your question because I haven't seen the data yet. It's gonna be, of course, that's what we want to know. But what the question is, what should we expect? Of course. So if we say that I claim here that cerebellar function is resilient to degeneration, it means we should find no correlation between the structure, the change in the structure of the cerebellum and the ability of the participant to perform, I mean, and the cerebellar measures, which it's always hard to show that there is no correlation, of course.
You do have to measure that there you do expect to see the correlation. So if you see correlation there and not in others, that would be very nice.
Yeah, I mean, I agree, but we haven't explored them yet. Yeah, so that's and of course here we've considered the whole cerebellum, and we are planning to look at some areas more specifically. So I mean, there's still a lot of work to do.
That's my next question.
Shlomi,
Thank
please let me go ahead and go to Will. Will, please go ahead.
Thanks, JJ. First, you are a great explainer. You explain very well, and the presentation, is very comprehensive. So me, as a learner, it's really a great opportunity. So thank you. It's really a learning opportunity. 2 questions regarding the anticipatory eye velocity effect. So if I understand correctly, your explanation for this effect, it's basically the repetition.
Yeah.
Correct? So what would be your prediction if you look at different blocks of the experiments?
Yeah, I mean, there's a huge literature on this anticipatory eye velocity. Even if we can't predict what's the future direction of the target motion, we'll try to anticipate. So we really, in that type of task, we are really anticipatory machine, and we really try to, to start to move the eyes before the target actually starts. So we haven't looked at block per block because you need 1 or 2 repetition to actually show anticipatory eye velocity. It's very fast.
Okay, and the second question regarding the speech adaptation task, what do you think is being learned there? What can you explain the mental process of being learned there?
Well, I think that there is a discrepancy between what you think you should hear and what you actually hear, because we modify the auditory feedback. So it's like for the visual motor rotation, there is a discrepancy between what you hear and what you are expecting to hear, and that's why this difference is driving the speech adaptation. That's the explanation.
No, I understand the discrepancy, but my question is, what is being learned? What is the cognitive mechanism being changed or being adapted during this task?
Well, I don't know whether there's a cognitive mechanism. It's
Or-
the sensory motor mapping that is adapted. You think to produce a sound, you will activate your muscle differently because you hear that the activation of your muscle, as you did before, do not produce the right sensory consequence. So you're gonna change the sensory motor map.
Thank you.
Thank you, Will. Rich, please go ahead.
Yeah, that was really fun tour there. JJ, so since we have Maurice already raising this question, I figure I'd like to hear more about it because we've struggled with this issue about on the mental rotation, a priori we were thinking that the rate was, the variable of interest, the slope, but we too had like, about a 300 or 400 or 500 millisecond intercept effect. And we know from other studies we've done that that's, you can't account for this as just sort of slow movement speed. If you do like a simple RT, the people with cerebellar degeneration are slower by like 50, maybe 100 milliseconds at most. So I'm just wondering, we struggled with trying to think about what to make of these intercept effects, and you really see it magnified in your study when you go from the older to the old. So
Yeah.
any thoughts about what to think about that?
Well, for me, there are really two different parts. Because when there is no rotation, I mean, it's a simple decision-making process. There is no update of mental representation when there's no rotation. The letter is flipped or not flipped. And so that's for me, more all the decision-making processes that dictate the reaction time for this. And this is the reference. And on top of that, for the other trials, you add the mental rotation. And so where does this difference comes from, I don't know, because I mean, there are many things that can explain this. But I think here the difference in slope for me is a better marker if we really think that this is, updating the mental representation continuously during the whole rotation.
Yeah, I mean, just to make the point that if we compare the older with people with cerebellar degeneration, you do get an intercept effect there too, right? So
yeah, no, that's right. And actually, our data here for the old participants, the slope is 4, which is very close to the slope that you had on average for the cerebellar patients. So we're in that range.
yeah.
But I guess I don't want to pile on, but I mean, you're saying that the zero rotation reaction times is decision-making process that, I guess implications is not so cerebellar dependent. But I guess the reason I was asking is because, in Rich's study where he looks at people who are, pretty specific cerebellar degeneration, that there's a massive difference in the effect size of the difference, like in terms of like statistical power for the difference is just much greater. In that study. So it seems like it is specific to the cerebellum, right?
Yeah, I mean, that's certainly what, we don't have any explanation for that. I mean, we that's right.
Yeah.
So
yeah, what we don't know is how much of the difference in reaction time at 0 degree here is due to the same processes that drive the difference between the controls and the cerebellar patients. But I mean, I'm not disagreeing with you, Maurice, on that.
Okay.
And so, Maurice, do you have any thoughts about what this difference is on what we'll call the decision-making components?
No, I mean, I'm not the scientist you are, so I don't know.
Thank you, Rich. Thank you, Maurice. Yu Chang, you have your hand up.
Yep. So hi everybody. I'm Yi Chen from Columbia University from Shenhan's lab, and yeah, it's a very wonderful talk. And I have two questions. The first is, we all know that cerebellum leads to motor part and non-motor part. So I really want to know, do you think that the cerebellum reserve that kind of do different ways for motor part and non-motor part? Because here you show most of your tasks from motor part, but I kind of little bit believe that there may be kind of difference in agents that the cerebellum reserve play different roles.
I mean, I don't know whether I have an answer to your question. I mean, the reason why we tested this mental rotation task is to see how far we could go in you know, because I mean, implicit adaptation, we knew we wouldn't see a difference because we have done that several times. I think we wanted a little bit to explore how far we could go with that idea. And I would have expected larger differences with the mental rotation, the slope of the mental rotation between young and older participants that we found. So for me, it was quite surprising. Whether the really, you know, I use the term motor reserve, but what is it exactly? That's very complicated to actually define. And even so, of course, it's taken from the idea of cognitive reserve, which has been established a while ago. But even there, it's the definition of what the reserve is. There is a little bit, you know, it's a bit vague. And so it's hard for me to then answer your question about whether the reserve would be different for cognitive versus motor function, because how do we actually operationalize the reserve is a very complicated thing. So I think we need to be very careful. I can't really answer that.
Yes, thank you. And the second question is, in your study design setting, it's very good. You have young, older, and a very old group. But do you think there's a break point that maybe in the young, older, they compensate very well, but in certain times the point and it's breakdown, so the older people, they cannot compensate well. So maybe the motor or cognitive functions will be break down, and the slope will be different from the young.
Yeah, I mean, when you design a study, you have to make choices. So actually having two groups of separate groups is more powerful than doing a continuous sampling across all age. That's why we did it the way we did. You win 10-15% power by comparing two groups than by doing the full age range. So yes, we don't know whether cognition, I mean, whether for any of those tasks, when there is a change, whether it happens at the same age and so on, but that would require much more data than what I have.
Thank you.
Thank you, Chen. Jessica, you have your hand up.
Yeah, hi, thank you. This is a wonderful talk and really beautiful set of data to look through and hear about. Super excited to see how your subsequent analyses move forward. So one of the things that I've just sort of been mulling over and thinking about, and Yicheng's point kind of brought this to mind as well, is maybe with respect to how inputs to the cerebellum, when we think about be it cortical or spinal inputs, may be impacting these concepts of reserve. And I was wondering if you had any thoughts on the degree to which how that might come into play. You spoke a lot about gray matter, but we are also white matter components here as well.
So we have also a diffusion weighted data. So it's our dream to be able to look at the peduncles from the cerebellum to the cortex and see how they are affected by aging, because there's absolutely nothing on the literature about that.
I know,
which is quite
astonishing.
But I understand why, because actually from what I've seen so far, it's quite tricky to do. So that's why, but we have a very high quality data. And then we also have resting state fMRI connectivity, because we wanted to look at these connections from what we have seen so far, but it's very, very early days. The strength of the connection between the motor cortex and the cerebellum seems to be slightly larger in older people. So that again seems to be maybe, and that we don't find that when we look at the connection between the cerebellum and more cognitive areas. So maybe for Yicheng, that was maybe a part of the story. We need to see whether this is true or not. That we have still plenty of analysis to do, but at least that seems to be, there seems to be something interesting there.
Yeah, I think that's really fascinating. I'm really excited to see it. We recently published some stuff where we show higher, actually cerebellar M1 connectivity. So I'd love to see this, with what you have and how this replicates across samples.
Yeah,
Some
yeah.
really exciting work. Thank you.
Ah, yeah.
Thank you, Jessica. Florian, please go ahead.
Hi, JJ. Great talk, really. And I like your distinction between older adults and old adults. Made me feel good. What I found intriguing that when you said with the eye movements that you couldn't measure those in the old adults because they're dropping their eyelids. I seem to vaguely remember that, eyelid control, supranuclear regions are involved in that. And I also remember that there are projections from supranuclear regions to cerebellum-peduncle. So there is something in that itself that they can't do that with respect to the cerebellum, possibly.
Yeah, I mean, we haven't tried that hard with the older old. It's simply that with the older group, we already had so much trouble getting good data and we needed to make the experiment shorter for the older old group because it was a little bit too much for them. Yeah, because we had trouble with older group, we didn't really try that hard with the 80 plus group. So, I mean, I don't know whether, we can make any inference from that. It's also, we don't have, it's the eye tracking in our key arm, so it's not the ideal eye position and so on. So there are lots of barriers to good, to have good data that are, I think, more technical than neuroscientific.
Thank you. Thank you.
Thank you, Florian. Maurice, please go ahead.
Hi, Maurice. You are muted, Maurice.
So we can't hear you, Maurice.
Yeah, we can't hear you.
Sorry about that. This is going to sound a little similar to my other question, but it's really not. So could you go back possibly to the slides about the Wolpert's force perception task?
Mm-hmm.
So there, I'm curious. I don't know why I had never noticed this before, but there's also a weird. If you go back to this, like the Wolpert data, actually, the previous slide, possibly. I don't know.
Yeah.
Yeah. So it's weird that there's a big offset effect here, seemingly, that seems to be as big as the slope. And if you calculate out the slope increases only actually like 1.2 or something like this, maybe 1.3, and there's a big fat offset. But of course there can't really be an offset here, right? So and it's hard to imagine that if you really presented like say 0.1 newtons of force, people would somehow match it with 1 Newton or anything like this, right? So it would seem like, and there's just a hint of it, I guess, at the 0.5 Newton place, that what would have to be going on here, just from common sense, would be that there's an initial sort of a high slope, and then and sort of an asymptotic so some kind of saturation type effect or whatever that's more modest, which is being shown in the fit to this data here, right? I think you probably would have to agree on that. And so I wonder, right, whether whether doing this task over small forces, maybe up to 1 Newton or even up to half a Newton would give you information that that was different and perhaps greater acuity. That's the one thing. The second thing was if you get back to your data, I mean it's a little curious, and I understand what you did and it makes sort of sense or whatever, right? But it's a little curious that that when you actually look at the data from the direct force matching class task itself, I mean the slopes really are, visually at least sort of like identical between the groups, right? And the differences that you're generating are because of the, I guess, the control that you're subtracting out. I don't really have any issue with the control, and I think it's reasonable, but I wonder if you had any explanation just why they might be so similar. Things just a coincidence that you know, that you really need to use this control data.
Yeah, I mean, I know Wolpert didn't do that, but for me, the fact that in the slider case there are differences. Well, for me, the right comparison is to do the direct minus the slider and use the slider as a reference.
So you
in
think it's just
Wolpert's,
coincidence that the slopes are so similar? I mean,
yeah,
I don't have any
mean,
problem
yeah,
with what
I
you're
think
doing
it's a
from
coincidence. Yeah.
first principles. But I mean, you know, you have to look at the data, and is the data, questions the data tell you something?
Yeah, I mean, I've no I think it's because at least in the direct condition, they seem to be to scale the force with the target force in a similar way. That's why what the only thing I can do. I mean, I don't have an explanation of why there are similar. I mean, I would not expect that they are very different. It's more the shift that is of interest for me between the two curves than the slope. The slope in the slider condition has been taken as a measure of how sensitive people are to the different forces, and there seems to be a small difference with age. But yeah, that's what the data
And I'm
tells
just
me.
curious, just because you probably thought about this much more than I. If you go back
Yeah,
to
I mean, it
what we did,
varies
what
so
do you
much.
think
That's
about what do you think
also,
about low forces? Like, you know, what's going on at low force levels? Like, but of course, you only look. Your minimum was 1 Newton, and Wolpert's minimum was half a Newton. But
yeah.
I'm curious what you think about what's going on with sort of force matching at low forces,
Yeah,
below Newton or below.
I really don't know how. Yeah, I mean, I didn't think about it. We've done an experiment, similar experiment with the whole arm, where our lowest force was 2 Newton, but it's for the arm. So of course the arm muscles are much more powerful than the fingers. Yeah, it's
My point is that the slope of the effect below 1 Newton is almost certainly like much higher, right?
might
By
be. Yeah,
double or triple the size of the slope effect,
yeah.
compared to what it is at
Yeah,
above,
I think
right?
we would need to properly test that and do really a range of smaller force and see whether there we see something different. I know there's just a paper out yesterday or the day before from Roger Enoka on force, and he has it's a aging paper on force perception, and he has a 5% of maximum voluntary contraction and 20%, and he finds difference between 5% and 20%, which is
Okay, and the
consistent
other
with
question,
what.
so you actually don't see an effect for young people, right? Which, you know, makes one
Yeah.
wonder whether there's something about, you know, you're somehow failing to capture here.
Yeah, well, so we did,
Sort of idea from Wolpert.
after a few participants, we did look. So the way we set it up, actually in the direct force for us, you were not pressing exactly on the lever, but 1 centimeter on top of it. And then because we didn't find the attenuation in younger people, we thought, well, maybe this is not ideal. And so for the rest of the people, we also did a condition where people press directly on the lever as here, and there we get the overcompensation in younger adults. So it's just the way we set it up, but it doesn't really change the story. It's just that for this, we have data from all participants, and from the additional control condition that for this exact condition, we have fewer participants. So it's just the way we set it up, but relatively doesn't change anything.
I don't understand. So on one hand, you're saying that if you do it with younger people, you get a different result.
No, I mean, just shifts
When
everything.
you press more directly, you actually see an increased
Yes.
slope for the young people.
But
you're
then
saying it
the
doesn't change the result.
overcompensation increases for all the groups. That's what I mean.
I see.
Then
By
even
the
you
same
do,
amount as each other?
I don't know by heart, but very similar amount, yeah.
Thank you,
So
Maurice.
you have more overcompensation for every age group.
All right, guys. Thank you so much. Will you let us go ahead and conclude, or did you have? All right. Thank you.
All good,
And yeah,
thank
that
you.
was wonderful. I'm glad you were able to explain for us the difference between your results and Wolpert's results. Wonderful, JJ. Thank you so much. Thank you, everyone. Hope you have a wonderful day.
Thank you, Reza. Bye-bye.
Good to see you.